Hello and welcome to Differential Discussions. I'm Melissa,
And I'm Dave.
and we're back with another body fluid. We're looking at
Woo.
CSF again.
Yay. CSFs, very critical body fluid.
Yeah.
And we're up on our low power and we're kind of looking around. So I see some red cells. I see a couple of like larger nucleated cells. A fair amount of red cells though.
Mm. Look at that big guy.
That is huge. That is not expected. Yeah, so there's some large mononuclear cells I'm seeing up in the corner here.
Ooh.
And this is perfect, right? So I try to tell students to kind of scan and look for clumps of cells. It's unusual for cells to be kind of stuck together. In body fluids, there's the plausibility that it's some kind of like a lining cell, tissue cells that are kind of in sheets. But very unusual to see something like that in a CSF.
Yeah. So let's just finish our scan and then we'll come back.
Yeah, definitely.
And then, like,
Yeah.
just looking how dark. Think about
High NC ratio, huge and dark. So my alarm bells are ringing internally, personally.
alarm bells are ringing. All right, let's go back to that clump.
Sure. Cool. We're on 40 or
40.
okay.
And pull it in. I mean, look at on 40, the size of these cells.
Yeah. I'll try to keep it in perspective with red cells here, right? It's a good kind of general tool. So red cells are 8 to 10 or 6 to 8, somewhere around 8 microns, and then you smush them, right? So the diameter will be a little bit bigger. So these cells are quite large, and there's a sameness. Honestly, this is
There's an oogliness. Yeah, that one's oogly.
that we need to see what the heck is going on here.
Let's pop down.
So large cells in the CSF, mono max are totally accepted and frequently seen in CSFs. So we want to be able to kind of, yeah, okay.
Wow.
So I
So
don't know.
these 6 cells take up almost the entire field.
Great perspective. Yep.
All
So,
right. So
yeah,
these are
I wouldn't even.
promyelocytes. Look at those granules. Right there, right? Right?
So yeah, I take your point. The promyelocytes certainly do have granules. You can also get like strange granules in cells that shouldn't be doing it either, right? So we talk about like automation and stuff in our field, and we're not going to have jobs. Just trust me for now. Chat GPT can't do anything about what's going on here. It's woefully inadequate. But excellent observation of those colored those chromatic granules there. The one that I want to just like kind of jump out of my seat over is this one. Is there a cell that you want to kind of start with, or
You can start with that one.
okay? So right away, I'm very troubled by the amount of nucleus to cytoplasm. So that NC ratio of 9, 10 to 1 or so, whatever the estimate might be, is typically owned by lymphs, right? I mean, there's no other real cell that has NC ratio like that. Of course, lymphocytes I expect to be just a tad bit larger than our red cells. This thing's gigantic. It's just huge.
They're just really big limbs.
Just really big. So then we talk about like the shape of the nucleus, right? So our lymphocytes typically have round to kidney bean shape. And trust me, normal lymphs will have weird shapes too. So we'll have some abnormal kind of lymphs, some abnormally shaped nuclei in normal lymphs.
Mhm.
Okay. But when we look at the outside of the nucleus, okay, so I'm looking at the contour. Something that is generally abnormal to find is kind of abnormal or herniations of the nuclear material through the nuclear membrane. So the nuclear membrane usually keeps things nice and tidy and neat inside, and that is not happening in this cell at all. We also have these weird vacuoles. We have deep basophilia, lots of color there, and then we have these spots in our nuclei that, I
One second
mean,
though.
sure,
Basophilia, we're talking about the cytoplasm.
yes, absolutely,
So
yeah,
when we say basophilia, we're talking about that dark blueness in the
yeah.
cytoplasm. Because I know sometimes people confuse basophilia with like basophilic granules, or what do you mean by basophilia? We mean the dark blueness in the cytoplasm.
Thank you for stepping in. Yeah, so I jumped around a little bit there, but yes, 100% what Melissa said. And the other thing that's bugging me is these spots in the nucleus
Hmm.
where we get that variable staining. Some places here, it's tough to spot where the nucleus even ends. This
Yeah.
does this spot a blue randomly in the nucleus?
Well, I think the other part too that's challenging is when you look at this, like right here, like you outlined the entire nucleus to look something like this, right? Originally. But when you look at it like this here, this part like right here, same thing with like this cell, like right here, is that granulation?
Mm,
Is that the nucleus? It's almost like you can't tell if that's nucleus or if it's cytoplasm, especially if you've never looked at like these large, ugly looking cells before. It can be really challenging to know, am I looking at primary granules like in a giant ugly myelocyte, or am I looking at part of the nucleus? And so that kind of lends it to the fact of you can't tell if you're looking at nucleus or cytoplasm.
mhm. And I always see this in laboratories where a tech is not confident or comfortable with what they're seeing, and they make more slides in a desperation to get it to look different. Okay.
Hmm.
I'm looking at this slide. These red cells look perfect. The staining on those red cells is bang on, right? I don't quite know what this is, right? But generally speaking, the staining characteristics of it are totally normal, right? So I can say with very high confidence, just from looking at this field, that my stain is behaving appropriately.
Yeah.
Okay.
I mean, the other thing too is you can look around, right? So we could
Yep.
move fields and we could go and find a normal, well, hopefully we could find like a normal neutrophil or a normal lymph, and then we could compare the stain there too. I mean, this clump would not be hard to find.
Yeah. Right. Right. Yeah. To find again. Right. Yeah. I kind of feel like in body fluids, it's easier to find big landmarks than on peripheral bloods. Peripheral bloods tend to be tricky to find where you found something.
Yep.
But, you can usually go back. And so, we're doing a lot of Hymen and Harn right here. That's a cancer cell in my
Yep.
mind. That goes straight to pathology.
Yep. Yeah.
Um,
I think another thing that lends it to being more of a cancerous cell is when we talk about lining cells in the
mhm,
past, like we talked about it with synovials, we'll talk about it when we talk about the ascites and the pleurals and peritoneals and stuff. We talk about how they have like little blebs and how like you can see the interconnections between the different ones. So these have blebs also, but they are not nice, normal looking blebs like you would expect to find when you're looking at a lining cell. There's blebs, but like look at this bleb here. It's almost like trying to like blouson out the way like a megakaryocyte would with pushing out some kind of platelet, or like a giant platelet would with blousoning out pseudopods. And these are not megakaryocytes or anything related to that. These are big ugly cells. So it's kind of like these big ugly blebs, these polychromatophilic, for lack of a better thing. It's just they're multiple colors. They're interesting. They're not even
mhm,
blebs like you would expect to find in lining cells.
mhm,
So the they they're large. You could look at them and think, well, is it just a lining cell?
mhm.
No, because the blebs and because of everything else that we've talked about.
And we can follow that line of thought too, right? If we want to be responsible and we're in an area of our own ignorance and we say, is this possibly a lining cell? So the question then begs, what is a lining cell in CSF? So we grab the textbook, we look it up, and we see leptomeningeal cells. You're going to see, hopefully in your lab, you have some kind of an atlas or reference. And if we looked at leptomeningeals, they don't look like this. They're also quite rare
Yes.
to have leptomeningeals in a CSF.
Yeah. And you may
Um,
hear them called choroid plexus cells.
yep,
Same thing,
yep,
but they're the lining cells of the CSF.
yep,
So we don't call them lining cells. We don't call them mesothelial cells. We call them either leptomeningeal cells or choroid plexus cells.
yep,
But yeah, they don't look like this.
yeah. So this is a bit troubling. Um,
Yeah.
so I'm inclined for pretty much every cell here to just put them into. I tend to stay with like an other category and then get more specific when I get a better handle on what's going on.
Yep.
But the normalish looking one is maybe this guy, but the fact that it has this lumpy bumpy exterior, I'm just going to be safe and put it in the other category.
Well, it has the lumpy bumpy exterior. It looks like it has two lobes here. It looks like it's got this weird giant nucleoli that's making an odd shape. And when a nucleoli is not round and making an odd shape, that's usually more of a malignant nucleoli, because if it's a normal cell with a normal nucleoli, it'll be a nice round nucleoli. So if
Yep.
it's making a shape like this guy is here, like it's almost making like a not quite a mushroom, but it's got like a little bit blue sond out over here and a little bit thinner on the bottom. The
Mhm.
fact that it's not round kind of also lends to where it's probably a malignant cell.
Yeah. Well said.
All right, let's look around, see if we see anything else.
Yep.
We probably will see something else, given that when we scanned, we saw some other fun things. But for reference,
Hey.
we have a normal cell, and look how
Yeah.
small it is.
Yeah. It's a normal little mono, right? Just
Mhm.
kind of chilling. Yeah, looking at the coloration, it's nice and light. It's kind of colors we'd expect in a CSF. Got a neutrophil.
Yeah,
So we have normal hematopoietic cells. So we've kind of quality controlled the stain effectively now.
yeah. I was
What a
just gonna
stain
say
is
that.
stain.
Yep.
Yep. Another poly.
Ooh.
And, you know, so now again, like, what is this cell, right? Um, very odd to have like a flat edge, that little kind of jagged corner. Um, yeah, the nucleoli too. Yeah, what is that, right? So that variability in staining, the hetero staining characteristics, you get dark here, lighter here. That's weird. That's not usual.
This is also kind of weird
Yep.
too, internally, and it makes me wonder, like, is this supposed to be part of like the nuclear membrane? Is that granules? Is that odd cytoplasm, or is that just cytoplasm staining two different colors, which again is weird.
Yeah. Yeah. I mean, I like, my mind keeps going back to meso. So mesothelial cells tend to have this fried egg kind of look. And this is like a weird inverted fried egg.
Yeah.
Is that? um, but yeah, very abnormal, weird looking. Don't expect to find this in a CSF.
Yep.
So this is going into my other.
Yep.
Okay. Oh, yeah. So there's some degrees of uniformity between the cells, but there's some variability. This
Yep.
one kind of tends to match some of the ugliest parts of all the cells that we've been looking at.
Yep.
So I don't like how dark the nucleus is there. We have that granulation, that pinkish kind of weird granulation in the cytoplasm. we have weird shaped nucleoli that's
Yeah.
maybe tiny. The nuclear margins, like what the heck is this little thing kind of sitting
Hmm.
there? Yeah, so I other this fella as well.
And it's huge again.
Hmm.
Oh, oh. Yeah.
So good. I love the lymph, right? So if you desperately kind of try to make more slides to make this fella on the right go away, right? It's futile. Like these red cells and this lymph is all I need. That is totally well functioning stain. It's doing exactly what it's supposed to.
Yeah. But like this cell is ugly. The nucleus is really dark. It's hard to see the real characteristics in the nucleus, although it looks like there could be nucleoli, but it's hard
Almost
to
impossible
see
to tell.
the way the cytoplasm is like it's almost like it's not quite blebbing. It's almost like it's like I don't know, it's making like these weird undulations in the edges of the cytoplasm.
Yep.
And again, the way the granules are like crowding to the nucleus, and
Mhm.
then this I assume it's granules anyway, and then like the just it is ugly and dark and does not look right.
Yep. So the dark staining is an abnormal finding of the cell. So in case that point has like landed clear enough.
Yeah.
And then this weird nuclear contour.
Yeah.
So I mean, it's consistently just
Yeah.
bad.
And I think just looking at the chromatin pattern of the lymph will help to emphasize like this is what normal heterochromatin looks like.
Yep. Yep.
And so when you compare that to this big ugly cell, euchromatin is usually light and fluffy and like very nice and easy to see. And this would be heterochromatin. So like what is that?
What is that indeed? That is a pathology review.
It is. Right to the doc. We're just gonna say this large unidentified cell that is wonky looking. Maybe I'll describe it for the doctor and send it back to the path to review. And because when we're looking at body fluids, it's not just hematopoietic cells that we're worrying about here. So
Right.
these could be some kind of cancer from somewhere else that has metastasized to this CSF.
100%. And that would be my guess, right? If you asked me to bet the farm, that's what I would bet on. These weird granules, huh?
Yeah.
Really strange. I do like the nucleoli in this area there. Beautifully abnormal.
They are beautiful. The nucleoli are gorgeous in some of these. Like that one you pointed out. There's a couple over here that are like, there's almost like a nucleoli like doing this down here where it kind of looks like they're almost overlapping right here.
Yep. And normal nucleoli are typically circle and ovoid, and we give a little bit of space for them to look weird if the cell's being kind of pushed around, right? So if a cell
Mhm.
gets kind of squished, it might kind of change elements, but there's no excuses here. This is
Ugly.
yeah,
Yeah, I agree.
birds of a feather.
Yeah. Can't remember if I came from this direction. We're just gonna keep going this
You got a neutrophil
way. Cool.
next to another cancer cell, probably.
Yeah, again, you have that big, dark, ugly staining area here. And then it looks like a nice prominent nucleoli over here.
And someone might tell me, "Hey, that might be a monomac,"
Yeah.
and it might be, it's possible, right? But um, there's a switch that flips in my mind, and I'm not saying that this is best practice, but for me, when I've confirmed that there's something possible malignancy going on, cells then have to prove to me that they're a normal cell.
Yeah.
Like I want to kind of flip it back the other way, and
I agree because given the company this cell keeps with that ugly dark staining area, the nucleoli, it almost looks like it's bilobed. yes, the cytoplasm looks like it could be more monomachy. Like
Mhm.
the cytoplasm, given the other cells that we look at, the cytoplasm does look significantly more normal than what we've been seeing. But the the chromatin pattern, those ugly spots, the nucleoli,
It's a problem.
yeah.
Yeah.
Well,
Well,
I think this is a good spot to stop.
yeah, me too.
Okay,
Interesting
well,
slide.
I think that's all we have for this. So thanks for watching.
Yeah, thanks for your time.